he was presented. So very honored and excited that he was able to come out and share with us. Will is a principal investigator and a physician scientist at the Center for Systems Biology and the Krantz Family Center for Cancer Research and the Department of Radiation Oncology, Massachusetts General Hospital and Harvard Medical School. He leads a laboratory in spatial and systems oncology and is associate director of the Radiation Biology and Research Program at MGH Cancer Center. His laboratory is focused on studying tumor stroma interactions in cancer. A lot of high impact publications, received many honors, including a Rhodes Scholarship, a Young Investigator Award from ASCO, a Next Gen Star from AACR, Burroughs Wellcome Fund Career Award, and 40 under 40 in cancer, some rising stars and emerging leaders. So just really impressive in the Sir William Osler Young Investigator Award. So really, we expect a lot from you today. So, so I'd like to welcome Will and give him a nice Michigan welcome.
Thank you very much, Dr. Keller, for that over the top kind introduction. Definitely don't expect too much today, but I'm very excited to be here with all of you guys. Super impressed by what you guys have put together with the SCSAP program. I kind of wish we had something like this where I am located. Certainly keep up the great work, and hopefully I've already met some of you guys doing really amazing work. Collaborate and really move the field of spatial, not just in the discovery phase, but I hope as a physician scientist, we can ultimately translate a lot of what we find to our patients. I have a laser pointer. Oh, there we go. Okay, perfect. Okay, so I know from speaking with Evan and Vanessa that I think you guys represent a lot of different disciplines and backgrounds. So I apologize if anything that I go through is too elementary, but I'm going to give some kind of clinical background. And then I thought, I would spend about half of today talking about some recently published work, and then spend the second half talking about a bunch of unpublished work. I think some of which many of you guys are interested in perhaps doing in your own labs as well. So as a clinician, I mainly focus on pancreatic cancer patients. I treat all different GI cancers as a radiation oncologist. About 10% of my time, I spend the rest of my time in the lab. And one of the reasons PDAC I really gravitated towards early on when I was a medical student was just, some of the numbers are obviously very devastating. It's up there with glioblastoma in terms of most deadly solid tumors, somewhere between 6th to 10th in incidence, but somewhere between 2nd to 3rd in terms of mortality. A lot of research has poured into this disease over the years, but our 5-year overall survival is still only about 13%, which is incredibly bad. So that really motivates myself as a clinician and a researcher every day and the people in my lab. There's many reasons why pancreatic cancer is very deadly, some of which you can see in this diagram. So there's both cell-intrinsic reasons. It's almost uniformly KRAS driven, and until very recently, hopefully this will change now, but that's been very difficult to target. A lot of the other mutations, driver mutations, and various tumor suppressors are also particularly difficult to target. There's metabolic reprogramming, very desmoplastic, immunosuppressive microenvironment that makes it very hypoxic, and that makes radiation and chemotherapy very difficult to be effective. And then there's also something I'm going to talk about a little bit today. You don't see this in this diagram. You don't see it in most diagrams. That is the nervous system input. So this disease is particularly strong. In fact, some pathologists, I know we have some pathologists in the audience, estimate that 95%, maybe even 100% of PDAC actually will have this interaction between cancer cells and nerves, known as perineural invasion, that I think we're starting to recognize is very important to tumor biology. And I'll get into that a little bit with one of the examples. So several years ago, when I came into PDAC, this was the field of PDAC as an early fellow. Spatial was still kind of under development, certainly was not the way it is now and as widespread as it is now. So back then, when we wanted to try to understand how we could effectively molecularly stratify our patients, people would look at somatic mutations. That doesn't really separate patients very well in PDAC. Or look at phenotypic characterization, often at the transcriptomic level. So several seminal transcriptomic studies done in bulk, summarizing this diagram here, really converged on separating pancreatic tumors into a so-called basal-like versus a classical subtype. I won't get into the details. This is just to introduce you to the terminology, as it'll come up a little bit later. But these cell states or subtypes end up being prognostic and predictive on some level. So if you look at metastatic patients getting first-line treatment, the purple line represents tumors that are in bulk characterized as classical. The orange are basal-like. You see that the basal-like tumors tend to have worse overall survival. And then from this waterfall plot, they don't respond as well to multi-agent chemotherapy. So the first question that my team and I were interested in was when I saw the patients that we were enrolling in various clinical trials, and often by the time they came to us, they had already seen some form of cytotoxic treatment, usually in the form of chemotherapy. At our institution, we actually give quite a bit of upfront radiotherapy in borderline resectable and resectable patients as well. And almost all of these studies that I showed you that led to this basal-like versus classical distinction had been done in untreated specimens. So we wanted to know what cell states look like in treated disease. And we also wanted to improve upon the resolution of these bulk studies. So a couple of years ago now, we did a fairly large single nucleus RNA sequencing study, and we did a de novo molecular taxonomy classification of various malignant states. And we were able to recapitulate using consensus non-negative matrix factorization some of the states that had already been identified in bulk, like this classical state, the basal-like, we resolved into a few different distinct states. And then more uniquely, we identified this neural-like progenitor or NERP state, which if you look at these tumors after treatment versus untreated tumors, the more treatment a tumor had saw before we resected and analyzed the tumor, the higher the program expression for this NERP program. So this was intriguing to us. So I'm not going to talk about it today because it actually is not a non-spatial project, but one of my grad students has really dove deep into this NERP program, identified the master transcription factor, it's BLIS3 that drives it, has created mouse models with CRISPR activation, looked at what this state really does, and he's finding that it's got some stem-like regenerative properties, wound healing properties, really comes up very strongly in the metastatic and primary setting after cytotoxic therapy. And it turns out, hopefully we'll have this published relatively soon, while this arises post-chemotherapy, it also happens to be quite sensitive to KRAS inhibition. So there might be some light at the end of the tunnel there and rationale for combining neoadjuvant chemotherapy with KRAS inhibition. But I want to kind of take this in the spatial direction because that's what you guys are interested in. And with these molecularly driven cell states defined with dissociated single nucleus RNA sequencing, we wanted to put this in the tumor architecture context, right? So I think this is what you guys think about all the time, so I'm not going to belabor the point. But my team, grad students, wrote a little review for Cancer Cell a few months ago, kind of summarizing where we are with spatial oncology and both technology and approaches as well as translation in the clinic. And as you guys know, when we wanted to bring this into in situ biology, we looked at what was available in terms of spatial approaches, and you can broadly divide these into two categories. There's the sequencing-based approaches. I know many of you guys use Visium, for example, or Visium. We were in the early days of using geomics, very different approach, but it has some similar properties. And these approaches, again, as you probably know, tend to do better in terms of molecular plex. You can transcriptomics cover even whole transcriptome pretty easily, but often at the sacrifice of spatial resolution. Even techniques like HD that have technically subcellular spot size, being able to assign a transcript to a specific cell is still sometimes a challenge because of diffusion and other challenges with the technique. On the other side, you have imaging-based approaches like Xenium, like Codex, protein-based approaches as well, like the Lunaphore Comet. Here, you tend to have subcellular spatial resolution quite easily, but then it's a little bit harder to get your molecular plex up. So definitely pros and cons of both approaches, and I'm going to talk about our application of one of each category to PDAC and what we've learned preliminarily. Okay, so we started this several years ago. So at that time, geomics was still pre-commercial. My mentor, Aviv Regev, had a little center of excellence. So we were doing a lot of early FFPE testing with the geomics technology. I'm quite impressed by its FFPE performance. although the instrument itself performed very unreliably at the time. So what we did was, with the single nucleus RNA sequencing data in hand, we went to match tissue blocks and we did serial sectioning, and we did various protein and whole transcriptome analyses. Again, this is all published, so I'll go through it very quickly. This was not single cell resolution or anything even close, right? So this was our circular ROI. This was something on the order of the radius was a few hundred microns. But you are able to essentially do optical, the optical equivalent of laser capture microdissection, right? So you can use your molecular specificity from various antibodies to separately interrogate, in our case, the cancer cells from the fibroblasts from the immune cells, right? Each labeled differently here. And then when you go into each of these separate cellular segments, you can, for example, the immune compartment, you can deconvolve and get various relative proportions of different immune cell types. For the cancer compartment, for the CAP compartment, you can score them for some of those subtypes that I showed you earlier. And then you can start to do neighborhood analysis. So this gave us kind of an initial impression of what cell types were co-localizing with what other cell types. And this was informative, right? We were surprised to find that CD8 T cells were actually enriched with some of the post-treatment high cancer subtypes. We actually expected the opposite, and this ended up validating through various other studies. But what we were also interested in was to be able to do detailed cell-cell interaction analyses. And we felt like, while you could do that, I mean, you can do that even with dissociated single cell data with techniques like cell chat. And with this, you can do at the ROI level, various cell-cell interaction analyses. We felt like to really do a good job, right? We didn't really want to have a bunch of hits that we would then go to validate and they'd all turn out to be artifacts of the spatial technique. We wanted single cell resolution. So then a couple years after we were doing that geomics work, we had the opportunity to do some early work with the spatial molecular imaging platform on the same tissue. So a lot of these are matched single nucleus RNA sequencing, digital spatial profiling, and spatial molecular imaging. So using this technique, which has some shared features with the Xenium platform you guys are familiar with, we were pretty impressed with some of the early results that we got. So you can see in this particular field of view, immunostaining at the protein level, you can see the architecture of the malignant glands and the surrounding stroma. At the RNA level, when we pseudo-colored these individual transcripts based on their expected cell type specificity, that architecture was largely recapitulated. And then when you actually zoom in to these individual cells, you can see that some of the stromal cells, most of the transcripts detected are light blue for CAF-enriched genes. Most of the cancer cells, as part of these glandular structures, are expressing epithelial-enriched genes. So we were quite happy with the quality of this data. So we applied this approach. We had a roughly 1,000 plex panel, 30 spike-ins, specifically designed to help us annotate the basal-like, the classical, the neural-like progenitor, malignant subtypes, as well as various CAF phenotypes. And we applied this in gene expression space. We got pretty nice separation between the malignant and non-malignant cells. And then looking at this dot plot, you can see that by marker genes, we had pretty clean separation of the expected cell types in pancreatic cancer, as well as our malignant subtypes and fibroblast subtypes of interest. So in collaboration with Martin Hemberg's lab, we then developed a spatially constrained optimal transport interaction analysis. This is a derivative of some prior techniques, similar techniques, where we were able to leverage both spatial information as well as ligand and receptor gene expression information to basically generate what you see here, which is this spatially constrained cell-cell interaction score matrix, right? So if you look, the solution to this ends up being quite robust to cost matrix design. The particular one I'm showing here that we went with is the simplest one. Basically, the cost of transport of a ligand from a source cell to a receptor on a target cell is proportional to the spatial distance between them, inversely proportional to the ligand and receptor gene expression levels. So once you have this spatially constrained cell-cell interaction score matrix, you can do various statistical analyses. So one of our first questions was we wanted to see if we could identify, after neoadjuvant treatment of our patient pancreatic tumors, what interactions are enriched, which ones are depleted, and then we were hoping that some of these enriched interactions, maybe they would give us a clue as to what was driving resistance to chemotherapy. So this core diagram is representing the overall enrichment depletion patterns of interactions between various cell types shown here. And what really jumped out was the CAF source cell to malignant target cell interaction being strongly enriched after treatment. When you look at what these specific interactions are, the red dots on this volcano plot are enriched, and the ones in purple are depleted. On the depleted side, we saw some collagen integrin interactions, some Wnt signaling. On the enriched side, we had a bunch of hits. To narrow this down further, we actually applied multiple different ligand receptor databases that were published, right? Because that's obviously going to change your results. And the 4 that we converged on amongst the 3 different public databases that we used, 2 of the 4 actually were in the IL-6 family. And the importance of IL-6 in various cancers and driving EMT, driving chemo resistance, and some other tumors hadn't been directly shown into the best of our knowledge in pancreatic cancer, was fairly well known. So we were pretty excited to see that. So we did some quick validation. We're doing some more long-term validation now. But looking at 3 well-established human pancreatic cancer cell lines, when you treat them with chemotherapy, 5-FU is the backbone of FOLFIRINOX, which we use for most of our patients. When you give recombinant IL-6, in 2 of the 3 lines, you see a significant increase in viability in the context of chemotherapy. And we have some other evidence now, other in vitro organoid evidence, and hopefully in vivo mouse evidence soon that this is the case. So, this is kind of a small example of going from spatial kind of discovery-based data to potentially something clinically actionable, right? So, there are IL-6 monoclonal antibodies like tocilizumab, for example, used in rheumatoid arthritis that actually did not do well in the metastatic setting for pancreatic cancer. But now we're wondering, maybe in the neoadjuvant setting, in the curative intent setting, maybe something like this has more promise. We're obviously not the only ones looking at in cancer at multicellular interactions. This again is from that review my grad students put together, looking at all sorts of different ways people are leveraging spatial technology to look at cell-cell interactions in cancer. So I want to give a couple unpublished. The rest of the talk is all going to be unpublished work now. So I just ask that you guys keep this to yourselves if you don't mind. The first part is still going to be very biological. I want to talk about how we've leveraged spatial transcriptomics to look at a huge interest of mine, which is cancer nerve interactions. I think are pretty heavily understudied right now and very important to pancreatic tumor genesis and progression. And then the last part, I'm going to give a few examples of ongoing projects in the lab where we're developing/applying emerging technologies and applications. Okay, so spatial transcriptomics of cancer nerve interaction. So very quick, but let me just check my time here. Okay, so I still have like 30 minutes, is that about right? Okay, great, should have plenty of time. Also, I know I'm talking pretty quickly, so if you guys have questions, feel free to interrupt me. All right. So tumor nerve interactions. I first became interested in this field as a medical student. I was taking care of a couple pancreatic cancer patients who were having a very difficult time titrating opioids to not have them essentially be somnolent, but also control their visceral pain. Pretty much an impossible task. And I learned from now a long-term pathology collaborator of mine, Dr. Mino Knudsen, about perineural invasion for the first time. So I'll introduce that, I think, on the next slide. But couldn't do anything about it then, was doing other work, some single cell spatial work when the pandemic hit. And that gave me and my small team at the time an opportunity to really dig into this whole burgeoning field of cancer neuroscience and what was going on there. So this table on the left here from the Drapkin group from a few years ago really caught my attention. And a couple things caught my attention. One is you see all these different non-CNS tumors on the left and all the different types of peripheral innervation. And first thing that jumped out was how many of these interactions are undefined, right? So there's not too many areas of cancer biology where I think this amount of broad, not defined knowledge is the case at this point. And then the second thing that was intriguing to me was, as a GI radiation oncologist, I started looking at the GI cancers. I looked at gastric, for example. I saw, okay, parasympathetic input seems to be pro-tumor cluster. And then you scroll down to pancreatic, another GI cancer that I treat, it's minus. It has the opposite effect. So that was curious. So clearly it wasn't a one size fits all, different subtypes of innervation clearly having different effects. Looking a little bit deeper, it was also interesting because, I think there's a lot more evidence of some of these kind of hallmarks that we've outlined here in central nervous system tumors from work by Michel Mange and Frank Winkler and others, where they've shown actually electrically active synapses forming between neurons in glioma cells, for example. There's some evidence of this from our lab and other labs that this is happening with peripheral tumors too, but there's certainly electrical interaction, which is quite unique to the nervous system. But there's other things, right? There is paracrine signaling that can lead to things like stress adaptation. Alec Kimmelman's group showed that serine-starved pancreatic cancer cells will actually increase their nerve growth factor production, recruit nerves into the local vicinity to release serine to them and help them proliferate. There's also some interactions that lead to proliferative signaling in the cancer cells, immune remodeling interactions. Pain was something that I mentioned was how I first got interested in this. Even supporting metastasis, for example. So lots of potential interactions, very interesting. Perineural invasion was kind of the easiest entry point because this is the most histopathologically obvious manifestation of cancer nerve interaction, right? So I think the definition of this is slowly evolving because it turns out there's evidence that even if cancer cells and nerve are not directly physically in contact, you can have very strong nerve injury signals. Nisha De Silva's group has showed this recently, even hundreds of microns away. You see a cancer gland, a nerve nearby, you see that the nerve often shows very high levels of nerve injury signals, like CGM and ATF3, for example. But the most specific definition that I believe our pathologists still use today for perineural invasion is encirclement or invasion into the perineuron, so like intraneural interaction, you can see here. So during the pandemic, my team and I couldn't do much in lab, so we did a pretty broad pan-cancer RNA sequencing analysis across 12 different cancers. We had over 2,000 patients. Many of them were annotated for clinical pathologic features, including perineural invasion. And we found that across all these different cancers for both death and progression recurrence, perineural invasion was an independent negative prognostic factor. But what we noticed in looking at the literature was that most of these prior studies on tumor nerve interactions for pancreatic cancer, but many other cancers too, had a few limitations. One was they were generally focused on specific pathways that lacked cell type specificity, and then also spatial context, right? So if you're looking with a pathologist at a tumor that has one focus of tumor nerve interaction, cancer nerve interaction, I should say, but the rest of the cancer cells are nowhere near any nerves. And then you look at another tumor where there's proximity throughout the entire tumor, probably not the same underlying biology is going on, right? So definitely isolating the cancer cells that are interacting with the nervous system versus those that aren't in the same patient tumor would be beneficial and require spatial resolution. So this is a few years ago now. We're getting pretty close to publishing this. There's been a lot of functional work. I'll show you just a little bit of a sampling of that. But on the spatial side, we generated these 12 TMAs to do this spatial screen of cell type specific gene expression associated with tumor nerve proximity. And so we started this circa 2021. So whole transcriptome geomics was the first approach we used. We've now since done some imaging-based single cell resolution spatial approaches. But what we did with geomics at the time was we generated a whole bunch of different intra-patient matched ROIs where we had areas of cancer with nerve involvement. So this would be what a PNI situation would look like. Areas in the same patient tumor with cancer but no nerves, and then nerve controls, nerve but no cancer. And we wrote a custom script. You can't do this on the software that comes with the instrument, but a very easy adaptation allowed us to separately interrogate the nerve compartment first, then the fibroblasts within 100 microns of the nerve radially, and then 300 microns, and then the cancer cells within 100 and 300 microns. And we did a whole bunch of analyses on these, but for the purpose of today, I'm just going to focus on the malignant cells and one particular finding that we have started to functionally validate. Because again, I think, as a physician scientist especially, I definitely like to, I think the spatial results are great, but I definitely want to, as quickly as possible, see what can be validated biologically in functional ways, and then hopefully brought to the clinic eventually. So what we noticed when we looked at cancer cells as a function of proximity to nerves, we saw an enrichment, very strong enrichment in these light brown genes associated with the mesenchymal signature in our cancer. The classical genes were depleted. You can see in this volcano plot in cancer cells near nerves. And then the neural-like progenitor cells are actually a little bit of a mixed bag, but slightly more enriched. And in particular, we noticed, this is a GSEA plot that you're probably all familiar with. When we looked at platelet-derived growth factor, PDGF signaling, we saw a very strong enrichment in PNI regions, in cancer cells associated with PNI regions versus those not. So again, multiple things that you could go after, but this really piqued our interest because PDGF-D, PDGFRB signaling in particular, more so than the other PDGF chains, very strongly overexpressed in various cancer cell lines, has been shown to have a crucial role in progression of several cancers. I've listed some of them here. Promotes EMT, metastasis, angiogenesis, can be induced by reactive oxygen species, such as what you'd see after radiation or chemotherapy. There's known crosstalk with various mitogenic pathways, as you can see in this review I took this diagram from. And then potentially most importantly, from a translational standpoint, you could imagine therapeutically intervening on this pathway. So I'm going to maybe show about 20% of some of the functional work that postdoc and his team in my lab have done on exploring PDGF signaling. And we have evidence that it seems to mediate neural invasion in malignant cells. So what you're looking at here is a matrigel dome. You have TD tomato positive cancer cells. These are cancer cell aggregates. And the left here, there's a dorsal root ganglia in the middle. And on the right, everything is the same, but there's just no DRG. So this is the control on the right, experimental condition on the left. And what you're going to see as I start playing this video is on the left, you'll see these TD tomato positive cancer cells in the matrigel start to migrate towards this dorsal root ganglia, which is the source of sensory neurons from the spinal cord, and invade. And then on the right, in this control, there's no chemotropic gradient, so there's no directed movement. And then this is the time-lapse projection image on the left and right of these two respective conditions. You can pluck out the dorsal root ganglia at the end and do confocal imaging. All of this red here are TD tomato positive cancer cells that have made it into the ganglia and invaded. And then when we took our cancer cells and used CRISPR activation to engineer them to express either the ligand PDGFD, overexpress PDGFD, or the cognate receptor PDGFRB, we found that the ligand expressing cancer cell lines had a higher invasion index and higher invasion speed. Suggesting, again, we have some other evidence, but again, for the sake of time, I just summarize and say this plus a series of other experiments suggested that there's some autocrine signaling. So these cancer cells, even when we don't drive expression of PDGFRB, are expressing some level of the receptor we measured, and the ligand is driving autocrine neuroinvasiveness in these cancer cells. Looking at this another way, now we put these cancer cells are expressing M neon green now. Inside the dotted line is 70% matrigel. On the outside, the dome is 100% matrigel. And this is a radial invasion assay. And now we're looking at the cancer cell line that's expressing overexpressing the receptor. And when you apply, recombinant exogenous PDGFD, just at nanomolar concentrations, you see that there's a significant increase in invasion area with the cancer cell line expressing the receptor. There's to a lesser extent, there's also the scramble line does express some of the receptor. You can see from this flow histogram, there's less expression of PDGFRB as you'd expect. But you can reverse this increase in radial invasion of this receptor expressing line with SU16F, which is a small molecule inhibitor of the PDGFRB receptor. Very specific, the IC50 is 10 nanomolar. What about in vivo? So if you go into the mouse and you look at sciatic nerves, you inject, again, going back to the TD tomato model here, you inject these cancer cells and you give carrier control or SU16F. Again, this is targeting the receptor for the ligand. We found that there was significant reduction in longitudinal invasion of sciatic nerves when you block PDGFD, PDGFRB signaling. Now, what about on the nerve side? So the nerves, right, these are sensory nerves from the dorsal root ganglia. They're multicellular units. It's not just neurons. There's also fibroblasts. There's endoneurial macrophages. There's Schwann cells. So what is actually potentially responding on the nerve side, if anything, to PDGFD ligand and this signaling axis? So we were a little bit inspired by Richard Hwang at Memorial's work, published a couple years ago, where he was looking at MIA-PACA2 pancreatic cancer cells. He found that these GFP expressing Schwann cells, which form what he called these neurotracts that would allow the pink RFP expressing cancer cells to invade along. So it seemed like Schwann cells played a huge role in perineural invasion in the reciprocal tropism we see between cancer cells and nerves. So we looked at our own data. This is still very much a work in progress, but we had this PLP1. So PLP1 is specific for Schwann cells driving EGFP. And then we also counterstain for PDGFRB. And we found that in these nerves, you can see on the left here, these green Schwann cells, and these are also expressing PDGFRB. The other cell type that's also expressing PDGFRB are fibroblasts, which is expected. And then with flow, we're able to separate these populations out. So you can see there's a distinct potoplanin positive fibroblast population, PLP1 positive Schwann cell population. And then just looking at the Schwann cells, you see there is this population of GFAP high, PDGFRB high Schwann cells that we hypothesized were responding to the PDGFD ligand being secreted by cancer cells. So now flipping from the cancer cells experimentally to the Schwann cells, if you look at PLP1 positive, EGFP Schwann cells and do the radial invasion assay, we want to see would these respond to PDGFD ligand. And sure enough, when you give DMSO, this is the amount of radial invasion you see, same time point. When you give recombinant PDGFD, you see increased radial invasion. And then when you give recombinant PDGFD, as well as the small molecule inhibitor SU16F, you see that that increase in radial invasion is abrogated. And you can see that statistically over here. Going back to our spatial data, geomics data, and then on the left here, cosmics data in the middle panel here, we found that when you look at ROIs, that where the epithelial expression of the ligand is in the upper quartile, that was associated with significantly higher nerve expression of the receptor compared to when you look at the lowest quartile of epithelial expression of PDGFD. And we also saw a significant association between cancer cells expressing higher levels of PDGFD. This is just an example here shown in pink, and Schwann cells expressing PDGFRB. Spatially with single cell residue. So in summary, our spatial perineural invasion screen identified candidate genes that may facilitate cancer nerve interactions, including PDGF family members. PDGFD signaling mediates neuroinvasion by cancer cells, both in vitro and in vivo, it appears. Glial cells also express PDGFRB and are responsive to exogenous PDGFD. So our working hypothesis right now is that reciprocal PDGFD, PDGFRB signaling may play a role in mediating perineural invasion in PDAC. And then ultimately, we are, as more validation comes through, hopefully, we'd be interested in potentially pursuing PDGFD, PDGFRB blockade for therapeutic utility in our patients. Okay. Let's check the time. Okay. All right, I think we're doing well on time. All right, so I'm going to spend the last part. This stuff is all very much in development. So very not polished, definitely open to suggestions, feedback. Just a couple of the many directions that we're taking in terms of pushing the envelope on spatial technology, and in particular for my lab, how we're applying it. So we're thinking a lot about, ultimately how these are obviously very expensive studies. You guys are actually doing a lot more of these studies than we are. Most of all the studies I'm showing you are done on a single instrument in my lab for various projects. We have a little bit of a core facility that Dave Ting actually has started running, but definitely not pushing the types of throughput. But I think, if this is going to have a bigger impact, and then especially in the clinic, throughput is a big deal. I think sensitivity is a big deal. I think pushing molecular plex, I'm going to show you one example of that, I think is cool, and certainly has some niche applications, but I don't think it's going to be as big of a deal as sensitivity and throughput. Higher dimensionality. I spoke with some of you guys today about this. I think both temporal and kind of three-dimension physical space really important. I think there's work from like the Chung lab at MIT recently with Curve and eFlash and things like that, where without or Laura Wood's work with CODA and serial sectioning, ideally optical sectioning, but serial sectioning to kind of reconstruct larger and larger 3D blocks of tissue to really understand interactions in 3D and not just in 2D. I think would be very helpful. Adding the temporal dimension, I think, is huge. and happy to see the work that's happening here with that. Really, really cool stuff. Spatial multi-omics, I think other than, I think in the single cell world, sometimes I felt like we would just go for more because more is always better, it seems, or certainly maybe on grant applications or whatnot. But how much more are we really learning, or are we just kind of profiling everything? It's always something I worried about a little bit. But I do think that like spatial multi-omics, separate from if you don't have a specific question you're trying to answer in the most resourceful way, that may be fine if you're working with scant patient samples, right? I've really seen the value of this more recently, where you have a biopsy sample, or you have a very limited resection specimen because a lot of it has to be used for something else. If you can do spatial instead of dissociated work, if you can do multi-omics instead of single modality, potentially you can preserve so much more information that you might not even know what you need up front. And I think that has a lot of value. And then I think on the clinical translation, I think it goes without saying, all these high-plex discoveries, hard to imagine, and maybe some of my pathology colleagues disagree. From my perspective, it's hard to imagine that moving into the clinical realm directly. So I think, how do we translate this into liquid biopsies, surrogate biomarkers, low to mid-plex assays, right? IHC is a very good technique that pathology has been using for a long time. How do we distill these crazy expensive spatial omics studies into something that can be close to as cheap as H&E, So I think AI will. I'm not going to talk about that today, but machine learning AI approaches certainly I think will play a huge role. Okay, so as you know, my lab has a cosmics. I think a lot of these things apply whether you're using MERFISH or Xenium or any other emerging technologies. This is one of the examples where there are some applications where spatial subcellular resolution, whole transcriptome coverage can have some advantages, but it also has many disadvantages, right? So the data you're spreading, it's already sparse data. You're spreading out even more across 18,000 plus genes now. So we had to do some workarounds with this, but the data looks fairly promising, and you have specific targets that you can enrich. If you want to do more unbiased cell-cell interaction analyses, for example, right? Before all that work I showed you on 990-plex, you're limited by what ligands and receptors are represented in your panel. Not so if you're doing whole transcriptome. So we decided to apply this to this clinical protocol that we have going, where it's quite simple. These are resectable, borderline resectable pancreatic cancer patients planning to undergo neoadjuvant therapy before surgical resection. We put the biopsy and the resection specimen on the same tissue slide. And then here's an example of what we've annotated. There's residual tumor, there's hypertrophic nerves, we're generating globules, and then there's the pre-treatment biopsy. We put 2 cores on there. And then long story short, if you just do the whole transcriptome, you can see some of the metrics here. On a per cell basis, we get on average a little over 1,000 unique genes per cell out of the almost 19,000 targeted. And then in terms of transcripts, we're a little bit over 1,500, so not bad. This is all FFPE clinical samples. But what we found was that we struggled a little bit. Malignant, non-malignant annotation was not hard, actually. But some of the immune subsets, right? Like CD8 T cells, for example, NK cells are already a little bit hard to annotate. I'm sure you guys have experienced this at the RNA level. Protein level is a relatively straightforward, but they don't always express at the RNA level some of their cell type markers as highly. So what we did was we developed this technique where we integrated, go from H&E to annotate the ROIs, and we do concurrent on 2 consecutive sections, 1K and whole transcriptome. And that has helped a lot. I'm not going to show a lot of data today, really just kind of the overall technique. My postdoc developed multiple different approaches to do this, but in the end, this kind of 2-step annotation has been performing quite well. So we go from our single nuclease or single cell RNA-seq reference, identify anchor cells within the 1K SMI data. That works very well. We've actually already published this in that 2024 Nature Genetics paper. We generate a reference then, and then we do supervised clustering with in situ type, which was developed by Patrick Danaher et al. Over at Bruker now. And then we made some tweaks to this for this application. And then to move this into whole transcriptome, we use Harmony integration. This is the pre-integration UMAP. This is the post-integration of the 1K and whole transcriptome data. And then we do the label transfer, and we get pretty clean separation. When you go back and look at marker genes, it actually looks quite good for whole transcriptome data. We were able to use this, again, so many things you can imagine doing with this data. But one of the quick things we did a gut check on was I was curious if the basal-like subtype, we know that's enriched after treatment. These are matched. So all the stuff we published before were not matched within the same patient. For pancreatic cancer, it's actually pretty difficult to get a lot of pre-treatment biopsy tissue. It's usually used for diagnostic purposes, and we don't have much left. But in this study of 5 patients so far, all matched on the same section. And we were able to show, going from the pre-treatment biopsy to the post-treatment resection specimen, that we see a pretty nice preliminary enrichment in the basal state at the expense of the classical state. So that's pretty much what we expected from both the literature and our own data. Okay, so the next kind of emerging application for us is multi-omics emerging, putting proteo-transcriptomics together. As you guys are well aware, there's a lot of commercial efforts to try to do mid to hopefully higher plex spatial proteomics and transcriptomics in the same slide. As far as I'm aware, nothing is commercially available to do things at 100 plex protein, 1000 plex plus for RNA on the same section. So a couple of years ago, we started doing, the question that my fellow wanted to answer was, we were identifying all these malignant cell states, and we wanted to understand the interactions with the immune system, right? So pancreatic cancer is a tumor type that does not respond very well to conventional checkpoint inhibitor therapy. We were starting to see signs from our spatial data, our single cell data, that the different malignant subtypes may engage different types of immune evasion. So we realized, well, if we really want to explore this question spatially, the RNA-based spatial panels are pretty good at identifying the malignant subtypes, certain other cell types, but not the best at immunophenotyping. So we really needed protein level information. So we integrated on consecutive 5-micron sections, close to 1000 plex spatial molecular imaging to identify cancer cell states with Codex 96 plex. For immune cells, we use a QPath-based algorithm for segmentation and annotation. This is what an example looks like. So here you're seeing this is the whole slide protein multiplexed image. And then here's a few different areas. Even though people think of pancreatic cancer as like an immune cold tumor, it's actually not the case. It's very heterogeneous. You see areas of high lymphocyte infiltration, moderate infiltration, high macrophage infiltration, low lymphocyte infiltration. And then at the glandular level, we did a match across. Can't really do single cell matching reliably, but at the glandular level, we found it was pretty reliable. And I think visually, you can even see that it's not too hard to match up gland to gland between protein space and RNA space. So again, I'm just going to give a little preview of what we're working on. We're doing a lot of validation now. We have some interesting findings, but we first did a pilot of this on 4 patient tumors. You can see some of their characteristics here, and getting close to 1 million cells for each of the sections. This is the panel here. This is the Codex panel. And we also did this on TMAs for validation, and we saw some high-level differences between areas with where the malignant cells are expressing high levels of basal molecular markers. We saw active immune infiltrates, these lymphoid aggregates, which I'll show more on the next slide. It's actually pretty shocking how diverse these were throughout. And then more classical molecular markers associated with abundant macrophages and to a lesser extent some lymphocytes as well. This is like some of the diversity we were seeing. So across the 4 patients, huge range in terms of the number of immune aggregates as well as the composition. So you get some immune aggregates that look like more mature tertiary lymphoid structures. Some are very CD8 T cell high. This one here is very myeloid rich. Some of them have a lot of mast cells. And then this one here, this is neurofilament. Here's a nerve running right through it. So again, our cancer neuroscience interests, we're very curious about why some of these lymphoid aggregates, or immune aggregates, I should say, are very intimately associated with nerves, and some are not, and what the difference in biology might be. Quite a simple analysis that we were able to do here with this type of data was basically ask the question of differential malignant expression based on immune composition, right? So in this example here, in this field of view, showing the macrophage kernel density kind of in the grayscale in the background, and then the malignant cells are in green. And then we can do a DE analysis comparing cancer cells that are proximal to the immune cell type of interest versus distal, and notice various patterns. So this is doing that analysis on various immune cell types. for the malignant cell expression. And for macrophages in particular, and we're validating this on a large cohort, we're in collaboration with MD Anderson, we noticed that, for example, the cancer cells that were associated with macrophages had a very high mesenchymal score, as well as an interferon signaling score, right? So things like this come out very naturally from this type of integrated proteo-transcriptomic analysis. And now we have a bunch of immune-based hypotheses that we're testing both functionally and with larger cohorts. Okay, the last topic, I think I just have 5 more minutes left. I want to talk about another less commonly talked about element of omics, and that's morpholomics. So spatial morpho-transcriptomics, for the sake of time, I'm not going to motivate kind of why morphology is important too much, but I think both from pathology as well as just looking at cell culture and looking at how different cells being treated with a particular therapy that are resistant or sensitive look, or even just our basal-like cells, their morphology is incredibly different than our classical cells. So I think there's a lot of latent information in morphology that can move us towards kind of those more resource conserving surrogate biomarkers for a lot of things. So long story short, my grad student and I worked on this approach where we start out with cell lines. We're doing this on tumor tissue slice culture as well now, but we treat with a therapy of interest. So pancreatic cancer, we're interested in KRAS inhibitor therapy. And then we do phase holotomography. I'm going to show you a movie here. So this is an example. It's all label free, and you can see really detailed subcellular morphologic changes during treatment, during steady state, and combining this with post imaging fixation, segmentation, and then spatial transcriptomic readout. And this allows you to do paired single cell morphologic analysis. So if you look at the quality, this is cell line data. So the quality, of course, is better. Using a 1000-plex panel, we're getting an average of roughly 5,000 transcripts per cell and almost 500 unique genes, right, out of a total of 1,000. We're able to, without any barcoding, really cleanly separate out the pool of 16 different human pancreatic cancer cell lines. We're able to just separate them out very cleanly transcriptionally and do various analyses. So we've also done cell painting developed by Ann Carpenter lab at the Broad. So phalloidin is going to label your actin, paxillin, your junctional marker here. And what we noticed is that RMC 6236 is one of the pan-RAS-on inhibitors that are being explored in patients right now. And we saw for MIA-PACA2 lines and other lines this very distinct increase in cell area, increase in actin filament expression, for example, that you can see play out here in these various lines and also visually. So a lot of cool analyses that you can imagine. Also, you see things at the level of multicellular interactions. So we found that when we treat with in this case, Mirati 1133, which is a G12D KRAS inhibitor, we saw that the treated cells, the ones that survived, would actually form these cell aggregates, these clusters. You see from this density histogram here that the untreated cells in gray are much more found in singlets compared to the treated cells in light blue are found in clusters of more than 5 cells. And when you look, there are specific gene expression patterns that are associated with this. Glycolytic enzymes, lactate dehydrogenase, for example, enriched in the treated clusters. Integrin expression patterns look very different in the clusters. And when we went into the mouse and treated with the Mirati compound or to these orthotopic tumors, we also found that similar to what we were seeing in vitro, focal adhesion was one of the most enriched KEGG pathways in the KRAS inhibitor resistant cells. And when you look at dependency map data, for example, we found that ICA3, which if you go back to here, was one of the most enriched in the clusters we saw. It's a specific dependency of the high basal signature cancer cells, right? So again, I think for the sake of time, I won't go into too many of these findings, but just wanted to show some examples of where we think combining morphologic analysis and transcriptomic analysis can be very fruitful. So with that, I'd like to thank all of you guys for your time. Thank you, Wanissa, Laura, all of the SCSAP leadership, and all of you guys for inviting me. And I hope we'll all have a chance to work together on some really impactful stuff. And then obviously all the funding sources are patients and all the amazing people who worked on it. Thank you.
Thanks so much for a great talk. We have some questions you can start answering.
Amazing talk and amazing data. My question is that, you start, most of your experiments depended on like 2D pancreatic cell lines. And I was wondering your future, whether or not in the future, you'll be concentrating on organs where you can actually.
Yeah.
Would that be more precise?
Yeah. Yeah, no, great question. I think you're right. Like all the stuff I showed you with the morphology stuff, because not like some crazy tech dev project, but a little bit of kind of like method development and integration. It was actually tricky how to get the phase hole tomography to be compatible optically, right, with cosmics. That's why we chose to go with 2D initially. We are doing 3D work and actually living like tumor slice work. Where right now, most people are able to get these tumor slices to survive in culture, maybe 5-7 days reasonably. We're trying to push that to 10-14 days with some changes in technique. I think you're absolutely right. I think one of the reasons we start with human tissue as much as possible or mouse models when it makes sense is because of that physiological relevance. I think specifically for the morphology project, it made sense to us to start with 2D lines where I think we're already able to learn some things, but absolutely, I think tumoroids, co-cultures, I didn't show a lot of it. I didn't show it today, but for the neuroscience work, we actually do a lot of co-culture work, and that can be pretty useful. Thank you for the question. Yes.
I have two questions. One is whether or not do you recognize some change of the cellular cancer cells, like next to the nervous system? As you demonstrated those invading cells showing like mesenchymal phenotype, but do you see like more specific states of the cancer cells?
Next to the nerves versus.
Yeah.
That's a really interesting question. I didn't put it up because I'm not sure I believe the data yet, but one thing my postdoc was noticing, and there was actually one other lab, I think, that I'm aware of that saw something similar. So maybe it's true. The phenotype of the cancer cells immediately adjacent to the nerve, speaking on averages here, right? More mesenchymal, as you said, a little bit more of that neural-like progenitor phenotype that I described as well, not as strong as the EMT. But inside the nerves, the intraneural cancer cells actually look more classical, which is very interesting, right? I don't know, we have to do some dynamic assays to see like what's going on. Like maybe this is like some sort of transdifferentiation back and forth. We know like EMT, there can be the reverse, right? MET, once metastasis occurred and a tumor is forming in a new site, right?
Yep.
So I think that the reason for that, I'm not quite sure, but it may have to relate to like some metabolic advantages of the classical subtype, in particular, utilization of the specific lipids in the intraneural environment versus the extraneural environment.
Yeah.
Is what we hypothesize.
I think that's good. And the second question is, typically, like conceptually, we tend to observe in cancer, tumor cell kind of hijack developmental processes. I just wonder if in pancreatic tissue context, do you recognize any communication between like the pancreatic cells, communication with nervous system in the developmental systems?
In developmental systems? So you're saying like early, kind of modeling early tumor genesis, or okay. That's something we're very interested in, actually. You guys may know Anirban Maitro, an pathologist over at, he has a lot of interest in pannins and stuff. So we've been discussing collaboration and looking at that. I don't have anything too intelligent to say to you other than I think it plays a huge role, right? So like Jamie Solomon at Pittsburgh, for example, she did a study maybe 9 years ago at this point, where she gave capsaicin, right? And I don't know if you're familiar with the study. Yeah. So it kills sensory neurons, and showed that the tumors in the mice would not progress beyond kind of these pre-invasive states for the large part. So I think there's a lot of communication in those early stages. A little bit harder to look at that in humans. I mean, you can do it intentionally, right? But not a lot of patients with pannins are getting resections, right? So we have to look for incidental findings a lot of times, or stick with like IPMNs or something like that, which are more commonly resected.
I have a question for the RNA protein spatial study. What is your plan, or what would be the best way to integrate the RNA protein data and quantitatively like understand the relationship?
Well, quantitatively,
Yeah, not just imaging, but like
Yeah.
how do you extract it and then quantitatively compare between different samples for two omics?
yeah, that's a good question. I think that's something that I don't think that, I'd love to chat with you more about it. I don't think there's a really clear answer about, because there is so much slide to slide variation. So what we have been trying to do is to be more like intra-slide control, like within, like, obviously, like try to label everything at the same time. We're not doing really quantitative immunofluorescence there, because I think that would be a big stretch, especially if you're comparing between different slides. But I think relatively speaking, we kind of had too much trouble with annotating, right? So like depends on your question, right? So if we're trying to generate confident immune cell type annotations, we can do each slide somewhat separately. We not necessarily can use the same threshold for every marker across all the different slides, but we can also do certain controls, right? So we can use kind of lower plex assays, right? And more reliable staining or IHC on like a consecutive section as kind of our reference standard to look at certain areas on the slide and see, okay, how many CD4s do we see there? Or is it matching up with how we're annotating on the high-plex like codex data? And then ultimately, most of the things we're asking right now are more cell type abundance based. So as long as we're fairly confident in like, okay, this is the proportion of this cell type in this area, this cell type in this area, and we have them all spatially localized, I think those questions we can more confidently answer. But I think if you're really trying to use the protein data and compare expression levels between different slides, I think I agree with you, it's very challenging.
Yeah, very nice. The perineural invasion is really interesting, in particular to me. I'm curious about two things. One is the various varieties of CAFs. I'm wondering whether one of them is in any way associated with driving that phenotype. And then the second question is, do you see any evidence of abnormal pathways in glycosylation in perineural invasion?
Okay, second question, I haven't looked specifically, so I don't know. Be interesting to take a look at that for sure. The first question, we've mainly focused on the malignant cells, but what we are noticing is that the IL-6 expressing, the ICAPs that express IL-1, IL-6, they tend to be more closely associated with the more mesenchymal cancer cells, and then those are more associated with the nerve. So that trifecta seems to be what we're seeing. But in terms of really deeply exploring what those ICAPs, we call them, are doing with the neurons in terms of reciprocal interactions, and you've got still a lot to look at. But co-localization wise, it's more that subtype than the micro.
I have a question. Thanks for your nice talk. I'm just curious, the epigenetic mechanism for neural tumor interaction or pancreatic cancer. Do you have any progress about epigenetic stuff?
In that category, no. I haven't really had the bandwidth. I think it's very. Do you have a specific question? I'm curious what you were thinking.
Yeah, because so many biological phenomenon is actually related to epigenetic imprinting. So I feel like there should be some epigenetic regulatory mechanism for neural tumor interaction or neural immuno-tumor interaction.
Yes,
that's my assumption.
Yes, no, I agree with you. I think that's another layer that we definitely need to look at. I think the neural-like progenitor cancer cell state that I was telling you about, we've relatively recently identified, kind of the regulon that's like strongly associated with it, and we can, with a single transcription factor, kind of drive most of that state in a mouse model. There are seems like there's some very unique interactions between that malignant cell phenotype and nerves, and that might be a really interesting place to kind of study potential epigenetic mechanisms. So we know it's also a very dynamic state, it seems like.
Your interesting work looking at both transcriptomics as well as protein was going to lead me to ask you the question, can you actually come up with a conclusion as to how well correlated the two are? Your previous answer to the question here suggests that variation from slide to slide makes that difficult.
Yeah.
But I just want to ask the question anyhow.
Yeah, so when you say correlation, you mean like between RNA and protein space? on certain
Will
things?
one predict the
Yeah.
other or that sort of thing?
Yeah, yeah. So I think not enough data yet to be super conclusive. You know, we have 4 fully analyzed, and the TMAs that we're doing with our MD Anderson collaborators will, I think, shed more light onto it. But I think the not great correlation, as you might imagine. So like what we annotate in RNA space, especially some of the immune subsets, what we think is like, you know, some subset of T cell, for example, like the markers when we look in protein space in that area. Now we're not doing cell to cell mapping, right? Gland level. So like the best we can do is kind of like, you know, a clump of cells in a certain area. Like what is the composition we're seeing there confidently at least? We're not seeing great matchup on that.
So if you had to guess, or maybe just bet, is there more variability on the transcriptomic side or on the proteomic side, or is it hard to tell?
Probably the correct answer is hard to tell, but I overall, for various reasons, trust the protein data a little bit more, even though we're at like high-plex. And I mean, the antibodies are pretty well validated before they go into the panel. And we've done it both on Codex and Comet now. And I think there is more concordance between the protein platforms. Not that that answers your question, but I think on the RNA level, especially with like fairly sparse, you know, we're doing SMI, right? So the close, you know, right? Like, I think that there's many things that there's so much dropout in like certain important marker genes for these immune cells that express many copies at the protein level, but far fewer at the RNA level, that I think that stochastic